of Physics, gives a talk to familiarize young minds with the topic and ongoing research in it. It is conducted in the affectionate memory of the late Dr. R.C. Popli, lecturer at St. Stephen's College from 1971 to 1994. I request Dr. Abhinav Gupta to tell us more about it.
So I think I must be the last one, Dr. Pope, and the last batch. So I was here from 1990 to 93. That was the last batch that Dr. Pope taught. I kind of struggled because he was quite sick by the time he taught us. And just a few years after I graduated, I found out. So just a few months after I graduated, I found out he had passed away. That was quite shocking. So he was struggling with cancer, and his struggle in a sense was quite inspirational for us because he persevered with his teaching and just wouldn't stop teaching even towards the end of his life, in the last few months of his life. So Dr. Popli has been an inspiration for an entire generation of students. So apart from his obvious skills with physics, what struck me most was what an excellent human being he was. So when I was in first year, we were in awe of him, and usually only the most serious students would go to him for advice, since we had the impression that he did not suffer wounds lightly. So later on, we eventually found that he had a delicate sense of humor, and it was a very compassionate person. So I remember we had a senior, and for several, we just couldn't get enough of Dr. Popli's classes. And he fell very ill for several days. He was seriously ill. And when he returned, it turned out that every day Dr. Popli would visit him in residence and take care of him. He had never appeared for any of his classes. Right? So this was the kind of person Dr. Popli was. So Dr. Popli is still fondly remembered by students. So he's probably the last ones around, I would say, who are now probably older than him when he was when he taught us. I still remember him as a wise physics sage whose eyes were always full of compassion. And through his latest series, he still inspires us.
Thank you, sir. So these lectures will be delivered by Dr. Shaon Chakraborty, who is an alumnus of St. Stephen's College and IIT Kanpur. He went on to do a PhD in theoretical biophysics from the University of Maryland College Park, where he researched on understanding the force response of biomolecular complexes using approaches from non-equilibrium statistical physics. And he then moved on to a postdoctoral position at Harvard University and Dana-Farber Cancer Institute, where he started working on theoretical models of cancer and evolutionary medicine. Towards the end of his stint, he transitioned into wet lab, where he used a variety of microscopic techniques to more broadly study cellular proliferation. He is now working as an assistant professor at the Simon Center for the Study of Living Machines, National Center of Biological Sciences, CIFR, Bengaluru, where his lab combines approaches from physics, statistical inference, and machine learning, as well as wet lab experiments to study cellular proliferation, the circadian clock, and the links between the two oscillators. Welcome, sir. And we also have Dr. Apurva Jayaraman, who is also an alumnus of our college, and also we have Anuradha Saha, who is also an alumnus from our college. Welcome to you too. I now request our principal, Professor John Lawrence, to inaugurate the event and present the bouquet to our speaker. And
Dr. Chakraborty, on behalf of not only the physics department, but the entire college, I'm very happy that you've agreed to come here and share your knowledge and wisdom with all of us. Thank you very much. This is from the physics department and from the college.
thank you.
With your permission, I would also like to welcome into our midst former students of college, including a surprise visitor who has dropped in without any prior knowledge of lectures happening. And he also happens to be from physics. So it's a very pleasant coincidence, if I may call it that. Let me, with your permission, these may not be bouquets and they may not be as big as the bouquet that we presented to our distinguished speaker for today, but they are special because they are from college. For you as well. Colleagues of the department and my dear universities. The traditions of college are extremely important. We have academic traditions, we have traditions in the residence, we have traditions in the college. The Popli lectures are important because, you know, they continue the academic excellence that the college has represented, not only now, but across time. And our speaker today is a fine example of the importance we attach to academic excellence. And like it is mentioned in your brief description, it is trying to take the excellence which is there here to the world outside. Because it's not just enough to do well in class and to get good grades. You know that college is outstanding, whether it's former students or whether it's teachers. College is outstanding because we must make a relevance to the world outside. So whether it's cancer research, or whether it's technology, or whether it's writing a book which captures the difficulties and the problems that people outside face, you know, that is the unique property that this institution has. I want to congratulate all of you for continuing this very good tradition. I want to very gratefully acknowledge Dr. Chakraborty's time and presence here. I want to congratulate all of you for being here, for paying attention, and going on to eventually, when your time comes, make a difference to the world. Thank you very much. Welcome.
Thanks a lot, sir. Uh, Without further ado, let's dive into the world of biophysics. I request do not use mobile phones during the lectures and put it on mute. Kindly do not interrupt the lecture. We'll have a question answer round at the end of the lecture. Thank you very much. Sir, you may.
Okay, can you guys hear me at the back? Is this loud enough? Yeah. This is better now? Yeah? Okay, good, great. All right, so thank you very much. And I have to say, this is really surreal to be here 17 years back, 17 years since we left college, and to be here on the other side giving the Popli Memorial lecture. It's really surreal. Um, I have these distinct memories. It almost feels like yesterday when I was there where Benisha and Nadia are sitting, introducing the Popli Memorial speaker. I think this was in 2007, and it was Professor P.M. Arora who had come that year, and I was introducing him. And so these memories are really fascinating. I remember we were making the posters, and both my co-conspirators in making these posters are sitting right there, grinning widely. And I think we also managed to bunk a few classes in that context. Uh, and so, yeah, thank you very much to everyone here for coming. thanks a lot to Professor Harish and the Physics Society for this invitation. It's a real honor for me to come back here. And also a very special thank you to both Nadia and Venisha. They've been incredibly kind hosts, and it's been a real pleasure coming back to Stevens after 17 years. And I hope I'll be able to give you some uh, insight into the kind of work that my lab is currently doing, and then hopefully excite some of you to think about biology, because I think often in physics we are not used to looking at biological problems enough. And over the next 3 days, I hope to convince you that this is really the age of biology, right? And you can bring in a lot of interesting physics, computer science, mathematics, machine learning to answer very, very interesting questions in biology. Okay? All right. So without further ado, I'm going to start. So I'm going to start and maybe I'll differ a little bit and say that let's keep it informal. If you want, just ask me questions at any point. It's okay. Even if I'm not able to finish my entire talk, that's fine. I would rather have you understand what's going on. Okay? So I want to begin by asking you a couple of very simple questions. Okay, so show of hands. How many of you find it incredibly hard to wake up early in the morning? You're just not able to wake up early in the morning. Wow. Okay, maybe about half the room. Okay, and how many of you simply cannot stay up late at night? I see the older people raising more hands on this question. Okay, good. So can someone tell me why this happens? What is the reason that some of us here are simply not able to stay up late at night, while some of us simply cannot wake up in the morning? What could be the reason? Any thoughts? Yeah, go ahead. Okay, very good. I hear the word cycle. Yes, there is some hormones involved in this, some cycles, but how exactly? Why is there a difference, do you think? Mhm. 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 Okay, very good. So hormone cycles, melatonin, anyone else? Yeah, very good. I mean, so habit is definitely a related aspect, though we will not talk so much about habit, but yes, good point. Habit also does influence our behavior. Anything else? A related question is, how many of you have traveled on an airplane and have experienced jet lag? Okay, some of you have experienced jet lag. Now, if you've traveled often enough, what you would have noticed is that if you travel from east to west, as opposed to west to east, it takes much longer if you're traveling from east to west to get back in shape. Why does this happen? And is there any relationship between the first question that I asked you and this? Any ambient light, but why not the other way? Why is one way much worse than the other way? Let's say all other conditions, you're not traveling north-south, you're just going east-west. So let's say sun is not such a, yeah.
I guess sound a bit stupid, but मुझे ऐसा लगता है कि the landmass is raised from one side and it's bit slanted from other, so maybe some sort of gravitation. I don't know.
Not really. It still has to do with cycles and so on. And there's a hint on the board. It's got something to do with the physics of your body's time. Yeah, yours. Mhm. Very good. So that's getting to the point, but why does that make this difference? It's related to that, of course. Um, so we, okay, yeah. So anything else that's different from these responses? Okay, good. We heard a few interesting answers, right? We heard about the sun rising earlier, later in different parts of the continents. We've heard about cycles, and so the main point is, yes, it is about cycles. And the next 3 days, I'm going to tell you about an incredibly important cycle that our bodies maintain, which is called the circadian clock, which allows us to maintain time. Okay, and it turns out that evolutionarily, this is an incredibly important thing that we have evolved with. Okay, so just a brief overview of what the next three lectures are going to look like so that you have a sense of what is coming. So the first day, which is today, I'll tell you about the past. Okay, I'll build up the context of what we knew in the past, how we thought about time of bodies. Okay. Tomorrow, what we are going to do is talk about present. So, what is the research happening on body time in the present? We talk about what exactly is the body clock. How do we measure these things? I was talking to Dr. Gupta just a while back, and the question is, how do you measure these things? How can you generate enough data that you can then start doing things like statistical inference and so on? So that's going to be tomorrow. And on the last day, this is the day when I'm going to talk more about my own lab's research. And here, what we are going to do is going to try to understand where the field is heading over the next maybe 10, 15 years, and ask from the level of organisms, can we go down all the way to a single cell and ask, does a cell keep time? Okay, and how do we measure that time if that's possible? And notice what I've done here is I've highlighted in green the kinds of physics that I will be talking about to explain these three different topics. Okay. So today, what you will see is a brief, a little bit of dynamical systems and how that can be used to understand circadian clocks. Tomorrow, what we'll be doing is going into a little bit of linear algebra and machine learning, and then on the final day, I'll try to introduce to you something called the physics of stochastic systems. Okay, there's something called the master equation and so on. And what I would love to do, and the way I've designed these lectures, is that every day I'm going to speak a little bit on using the slides, and then I'm going to move to the board, and I'm going to actually work out very simple examples that will be only a slight advance from what you would be covering in your 3 years at college. Okay, so I hope that all of you can actually take back something and really learn something new from these lectures. Okay, all right. So with that, I'll give you some context into the idea of circadian clocks and where it all really began. So it turns out that even over 2000 years back, if you travel way back in time, ancient civilizations, which included India, China, and all these other civilizations, we knew that there is rhythmicity constantly in the environment. Okay, this was our day-to-day lived experiences. We could see these rhythms, and in fact, there's a very subtle observation that Aristotle makes, and you can see that over here. He says that he's talking about nighttime bees and the sleeping behavior of bees. And see what he says over here, which is very interesting. He says that but the fact that insects sleep may be very well seen in the case of common everyday creatures, for not only do they rest at night time from dimness of vision. So he's thinking that you might think that you sleep at night simply because you can't see. There's no sunlight, you can't see, there's nothing else to do, so you sleep. But he says that, but even if a light is candidly presented, they continue sleeping quite as soundly. Okay? This is actually quite a profound observation because it's hinting at the fact that there are rhythms in the body that are distinct from whatever is going on outside. And you'll see that in just a bit, okay, why that becomes important. And of course, over time, people started realizing that there are rhythms in our bodies as well, which is 24-hour rhythms. And very common example, right? That whenever we fall sick, whenever you have a cold, you will invariably find that when you go to sleep is when it's getting the worst, right? So this was, of course, also observed back in the past by the ancient Greeks. And Celius Aurelianus says that he's talking about asthma, and he says that this is much more at night than during the day or in the spring. Okay. So clearly there are rhythms. These are roughly 24-hour rhythms that people have observed. But the interesting thing is that for over 2000 years, what people thought was that these rhythms are being driven by the cycle of day and night outside. Okay, this was a natural and obvious conclusion. You see 24-hour rhythms in the body. Your night is when you are getting worse. These are 24-hour cycles. And of course, there's the sunlight and day and night outside, which is also a 24-hour rhythm. So the natural connection is that because there is sunlight and sun and moon, there's a 24-hour rhythm outside. That is driving what's going on in our bodies. Okay, so that was the prevalent thought for over 2,000 years, almost 2,000 years, till 1729, where a very, very simple and fascinating experiment was done by this French person called de Maillard. Okay? So he did something really, really simple. He said that, well, if these rhythms in our bodies are being driven by the cycle of day and night, then why don't we take a plant? So this is a mimosa plant. This is a very common plant. There are Bengalis around here. It's called Lachapoti. I don't know if there are any Bengalis here. So this is an extremely common plant that we find everywhere. You touch it and it shrinks, right? What's that? It's touch me not. Okay, good. Not enough Bengalis in this room. So he took this plant, and what he did was, so these plants show a natural variation in their leaves. Right? During the day, they come up. At night, they droop. So he argued that, well, if this rhythm is really being driven by the external cycle, what if I just go and place it inside a dark cupboard? Right? What happens then? So he did that, and lo and behold, that rhythm still persisted. So this was the first time someone is suggesting that these rhythms are actually disconnected from the cycles that we see outside. Okay? But he was a good experimentalist, so he realized that his experiment had many flaws. Can anyone see what the flaw is? There's an obvious flaw in this experiment where you just put it inside a cupboard. The obvious flaw is that there are many other variables that you've not controlled for, right? For example, temperature. During the day it's hot, at night it's cold. So even if it's inside the cupboard, maybe it's the temperature cycles, which are also 24-hour cycles, that are driving this rhythm, right? So how do you really prove that this rhythm is inherent or endogenous in the plant? Okay? So it turns out that this is not an easy question to answer, and people struggled with this from 1729 for over 200 years to really prove what is going on over here. And here are two sort of extreme examples that I found. In fact, one from a group in JNCSR in Bangalore, which was a lovely study. Shiva Basu is right now my colleague at JNCSR, and they did this lovely study here where they grew Drosophila, this is the fruit fly, for over 600 generations in the dark to see if these rhythms persist, and they did. People also sent plants up in outer space to see what's going on, and invariably what you found was that these rhythms persist. Okay? So this is a very, very crucial idea that took us many years to figure out, which is that these rhythms are endogenous. And what I mean by endogenous is it's there within us. Okay, it's within plants, it's within animals, it's within us. Okay. So what's the connection with the day and night cycle? We'll come to that in just a bit. But the real evidence, right? So none of this was real sort of clinching evidence that these oscillations are endogenous. It turns out that the real evidence is actually rather simple. And it was already actually present in these very classic studies that a person called D. Control had performed already in the 1800s. So he had actually very carefully measured the time period of these oscillations when the plants were inside the dark chamber. And what he had noticed, and which had gone sort of kind of unnoticed for over a century is that this time period is not 24 hours. Okay? This, for some plants, is 27 hours. For some plants, it's 23 hours. So it's all over the place. So that, of course, begs the question, if these rhythms were directly connected to the day and night cycle, why would the time period be all over the place, right? If it was connected, it should have been 24 hours, but that's not the case. And this is very clearly seen in certain experiments that's been done in mice. Okay, these are very, very interesting experiments. What happens is that mice are what are called nocturnal animals. Okay? So mice sleep during the morning and they are awake and running around and doing activities at night. Okay? So what you can do is you can build what are called wheel running experiments. So these are real experiments that people do in the lab. What you do is you take the mouse, you give it artificial 12 hours of sunlight or daylight and 12 hours of darkness. Okay? So what this figure here is showing, each row is a particular day. So this is day 1, day 2, day 3, and so on. This is the first 12 hours when you have light, and this is the next 12 hours when you have dark. Okay? Now what you can do is when the mouse runs on this wheel, you can detect its motion by a kind of clicking mechanism here. Okay? So whenever it steps, there's a click, and you can detect that this mouse is moving. Okay, so these graphs here, these are called active graphs. So these black bars represent whenever the mouse has made a step. Okay. So using this, notice what's going on. In the first 12 hours when there is light, the mouse is sleeping. Okay, so there's absolutely no activity. The moment you turn off that light and it becomes dark, the mouse starts running around. Remember, the mouse is nocturnal. Yeah. So you see a nice 24-hour rhythm. 12 hours it's asleep, 12 hours it's running around. And this continues every single day. But notice what happens when you turn off the lights and put it in constant dark conditions. Okay? This is, once again, we are sort of going back to the kinds of experiments that De Mairan and De Candolle were doing with plants, but now with animals, right? You've put the mouse into a constant dark condition. The very interesting thing that you notice is now this activity pattern starts sliding earlier and earlier with every day. Okay? So can someone tell me, what would you interpret from these kinds of experimental data? What would be the first thing that pops out when you see this? Absolutely. Yes, very good. I heard all the right answers in the buzz. So there's a phase shift every day. But what that is telling you is that, what does it tell you about the time period of the oscillator inside the mouse? Is it smaller or is it larger than 24 hours? Smaller, exactly. Very good. So what's happening is the mouse has an endogenous rhythm which is slightly smaller than 24 hours. Okay, it's about 23 hours. And hence, if you keep it in constant dark, that rhythm is still going, right? But because it's 23 hours and not 24 hours, the mouse wakes up 1 hour earlier every single day. And hence it gets completely phase shifted from the cycles of external 12-hour light dark cycle by the time within a few days. Okay, so here it was 12 hours light and 12 hours dark. It was completely synchronized to the 12-hour cycle. Every morning, or rather every night when the light is being switched off, the mice are waking up exactly at that time and running around for 12 hours. The moment now you've put it into darkness, because its rhythm is endogenous, it's still running. Even though it's completely dark, something inside the mouse is still running, but it's running with a 23-hour rhythm, not a 24-hour rhythm. Hence, every day or every mouse day, it starts waking up 1 hour phase advanced. Okay? Does that make sense? So if this line was not like this, but it went like that, what would you then interpret? Time period is longer. And in fact, the fascinating thing is in the 1970s, the sort of the fathers of chronobiology, there were two gentlemen, they actually did this experiment on humans also. So they took their entire lab, right? I don't know if we are going to be allowed to do this in today's times. They took their entire lab and went off into these deep caves. Okay? They went off into these caves, no light, nothing, and they kept track of their daily activity patterns. Okay, so they lost complete touch with the outside world, and they realized that humans go like that. So on average, our circadian clocks are about, now there's some controversy about this, but roughly now what we believe is it's about 24 hours and 10 minutes. Okay, so there's a 10-minute, it's larger than 24 hours by 10 minutes. Okay, and in fact, this point is very crucial in trying to understand why jet lag is worse when we go from east to west. So keep this in mind and think about it, and maybe by the end of this class, I'll tell you what's going on. Okay? The fact that in humans, our average cycle time is larger than 24 hours. This is the crucial bit. Okay? Yeah.
So this has nothing to do with the rotation of the Earth. Actually, that in our evolutionary times.
So we're coming to that.
So
Yeah.
what I'm wondering about, has the rotation period of the Earth changed significantly over time? Is it possible that what you're seeing is a
So what
I'm wondering, I
yeah.
don't know, over such time scales, if it has.
Right. So what I'm getting at is that the connection with the outside world is simply that of a coupled oscillator.
Yeah.
So that's what I'm getting towards. Now, whether it has anything to do with that outside thing, I don't think it's very well understood. But the point is, in our bodies, right, we have oscillators of all kinds of time periods. We have so-called ultradian oscillators, which oscillate with, say, 3 hours, 4 hours time period. We have other oscillators that are even faster. During development, we have oscillators which are a few sort of hours, a few minutes also, right? So we have oscillators with time periods across the 24-hour time scale. What I think happened was we just had these oscillators, and I'll explain tomorrow that these oscillators are very easy to build from chemical reactions, right? So you have a bunch of these oscillators already lying around. It so happened that it was a 24-hour cycle outside, and that's what entrained one particular oscillator that already existed in our body. So that's how I think about it, right? But there is no clear-cut answer to this.
No, but what I'm wondering
Yeah.
is why not 24? I mean, the difference, would it have to do with the fact that maybe the rotation, because the Earth itself, due to the geological events,
That's also unlikely because, again, what I'm going to talk about, and the major point here is that there is a large variation in people, person to person. So on average, it's 24 hours, 10 minutes, but there's a wide variation between person to person, right? So some people may have actually 23 hours time period. Some people might have 26 hours time period, right? So then that argument is not as strong, right? So as I'm going to show you is what I think happened was you just had this oscillator which is close enough to 24 hours and close enough to be entrainable, right? To be within the range of entrainment. And this is a word I'll come to when I work out a little bit of the theory. Yeah.
I see a certain gap in this. Is there a significant?
Yes, very good question. I mean, you've got a very interesting point. There is a gap, and then you see this. We don't understand this very well, but some people think that on top of the circadian oscillator, there is something called a morning and evening oscillator as well. So there is some explanation for why these things happen, but it's not very well understood. There are explanations that there is a separately, there's some sort of morning oscillator and some sort of evening oscillator, which are approximately 12 hours potential. Right? So there are many oscillators in our body, and perhaps it's one of those that creates this kind of effect that you correctly picked up. Yeah, yeah.
It's a very random.
Yeah, go ahead.
Like in humans,
Mhm.
there's a concept of afternoon nap,
Mhm.
and it generally occurs again. It also occurs in a 24 hours gap. So similarly, like in this, the gap, what I was thinking, maybe the mouse rested a bit in that time.
It's possible, and it's
And
also, I mean, with humans, I mean, it becomes much more complicated, right?
like
Because after
it's,
we
it's
eat, and then we have these sugar highs.
yeah. But what my question was, the time period is like for the 12 hours it was running, the later 12 hours, and even if it is shifted, it is still running only for the 12 hours. Like the running time is still
Not
constant.
exactly 12 hours, right? Because it will be, the total is 23 hours. So it's slightly different from 12 hours.
I mean, the running time of the mouse isn't constant.
No, it depends on, because it's a whole thing is 23 hours, right? So then it's awake for half the time and running for half the time.
Okay, it would be slightly less than
It
12
will be
hours.
slightly less, yeah.
So the whole thing would get shifted, or even if it is dark, it would still run. Like the 11, like suppose it runs for 11 hours. So the 11 hours would get shifted to the left again, like constantly.
Right, like you see that's happening over here. Yeah. Okay, so yeah. What's that? So very good question. So why doesn't it happen here? That's what you're asking. So this is exactly what I'm coming to now, and this is also where you'll see that some physics-based theories from dynamical systems help explain that. So keep that in mind. We haven't really explained why this phenomenon is not happening here. You'll see that coming out of some very simple mathematics in just 5 minutes maybe. But so then the question is, if the endogenous oscillator is really not connected to the rhythms outside, then what is going on, right? I mean, why do we, exactly as we asked, why do we see this kind of behavior over here? And so this is now coming into the 1950s, 1960s, when people started realizing that these experiments, so how many of you know this person here? I hear a buzz, but I don't hear any concrete words. Who is this? Very good. I heard somewhere Huygens. Yes. So this is Huygens, the famous clockmaker. Okay. Um, so what people realize is that, in fact, this circadian clock system and the external day-night cycle is in fact very much like the coupled oscillator systems that he had described back in, I think, the 1600s. I might be getting the date wrong. But what he has shown is that if you have two clocks that are completely asynchronized from each other, and you couple it via a wooden beam, that's sufficient to set them to oscillate in sync. Okay? And in fact, this is exactly what is happening going on, and this, we've now begun to understand that this is exactly what's going on with the circadian clock and external rhythms, is that you have the circadian clock oscillations going on its own inside our bodies, okay? Not exactly 24 hours, which is very important, somewhere around 24 hours. But then you have the external cycle of night and day. And what's happening is in our eyes, we have photosensitive receptors that can detect this light. This information then goes to the brain. There's a particular part of the brain, it's in fact a tiny, tiny region in the brain. It has about, I think in the latest count, it's about 10,000 neurons in this region. It's called the SCN, it's the suprachiasmatic nucleus. And that is the master clock of our body. Okay? So this outside information goes in here and it synchronizes or entrains this oscillator over here. And you'll see just in a moment what entrainment really means. Okay? So you have a coupled oscillator system, And therefore, just like these coupled pendulums, even though their frequencies are somewhat different, they start oscillating in sync, okay, with the same frequency. Okay, so this is the other very important concept of entrainment by the sun, the day and night cycle of the internal body clocks that we have. Okay, so two important things that we've learned. One is that the oscillator system in the body is endogenous, right? It will run even if you keep the human being inside constant dark or in fact constant light conditions. So it keeps oscillating, but the time period is somewhat different from that of the external 24-hour rhythm. Okay? These couple and therefore you get what is called entrainment. Okay? So now what we are going to do is we're going to move to the board and we are going to see in a very simple dynamical systems model how one can explain this kind of entrainment behavior, in particular this kind of behavior. And as any good model should do, it should not only explain something, it should also predict something very interesting. Okay? And what we'll see coming out of these simple equations is also the idea of what I'm going to, is the phase of entrainment or chronotypes. And that we will look back and show, will explain why some people wake up much earlier in the day and why some people simply cannot wake up early in the day. Okay? All of this will come out from a very simple dynamical systems analysis of this kind of coupled oscillator system. Okay? Yeah, questions?
We see that the phase keeps on shifting.
Uh-huh.
If we keep the mice in the dark long enough, will it again become constant?
So if you keep the mouse long enough in the dark, it will keep shifting, and at some point it will come back in sync with the day-night cycle, but then it will again keep going, right? And in fact, what people have suggested is that in blind people, this is in fact what is going on. It's actually quite terrible. It's called chronic jet lag. So what happens with blind people is if they cannot receive any light at all, they are constantly, their times are shifting with respect to the external day-night cycle. So it's actually a terrible condition to be in because you're never in sync with the outside world.
The 24-hour cycle is fixed, but the day and night cycle is never fixed. So that should also be disrupting the internal cycle because of the conflict with the two.
You mean
The daylight at the same 12 hours, it's not 12 hours.
It's not exactly 20.
6 hours.
Yes.
Maybe winter, summer.
Absolutely. So those are again very interesting fields of research. So there are people who study seasonality, right? So you have longer days and you have shorter days when the seasons come. So how do these oscillators cope up with those kinds of systems? These are all questions that are in fact deeply asked. But I'm not going to go into this whole idea of seasonality, right? It will take us a bit too far from what I want to talk about. Yeah.
A continued question, like if we map the mice or a plant for longer days,
Mhm.
at any point will we observe any sort of random activities, something that is not periodic, what we expect?
Random activities.
Yeah.
What do you mean?
Like for the case of mice, you're saying that it's shifting by an hour every day
Mhm.
in this way, or like, so is it possible that we will see that it's not what we were expecting, but completely random, let's say?
So, I mean, that does happen, but what we are realizing now, that's more a disease condition. So, for example, there are ways of giving mice jet lag in the lab, right? So you can put them off these schedules by giving random lighting conditions, and there you can see that these mice will behave erratically just the way you're saying, right? In fact, this is currently a very, very hot topic in modern chronobiology and physics of circadian clocks, where one tries to understand if these circadian clocks go haywire, right? If their phase shifts go off, then what happens? What kind of erratic behavior one expect, right? So this is much more related to disease rather than sort of healthy mice. Mice show very, very typical behavior in perfect light dark conditions. Okay, that kind of erratic behavior only happens when, for example, you've introduced a cancer into the mouse or things like that. But it is a thing that we study in the lab.
Can we say the same for humans as well?
So there are no direct reports of that, but for example, there was a recent study of about, I think maybe 1,000 nurses. It's called the Nurses Health Study from Massachusetts General Hospital, where they looked at nurses who do night shifts. So they completely screw up their light dark cycle, and it's been shown that their risk of breast cancer is significantly higher than uh, normal people. Right? So once again, if you screw up this connection with the day and night cycle, then you get erratic behavior. Okay, so that's largely what the field is thinking right now. Yeah.
My question is, why don't we consider the possibility that at the time of evolution, the living organism picked up the cycle from the day and night thing or the 24-hour cycle and then adjusted it according to its habits or the limitations it was facing, and it keeps continuously checking it by the day and night perceptions?
Mhm. I mean that is possible, but again, it's very hard to rule out what you're saying, and I think this also goes back to what Professor Gupta was asking. It's hard to completely rule that out, but the likelihood of that is lower simply because, as I was trying to argue, that different people have very different um, time periods, right? So why would you then have such varied time periods? It's likely that it's just the chemical oscillations in your body, and we'll see that tomorrow, getting synchronized and entrained by this 24-hour rhythm.
Why not to assume that the nature was so intelligent to make two oscillators, one inside and one outside, and let us just sync it and keep
So
designing the
maybe
nature?
we should not go into this idea of intelligent design. Maybe that's not the best thing to discuss over here. I would like to think that design was not intelligent. So these generally happen out of random chance events, and then evolution sort of driving selection on things that work better, right? So that's generally how I would like to think about things. If you had random different stochastic events, right? There was standing variation, and then you had evolution coming and then selecting upon that random variation that you already had. And it's not that someone intelligently created something here, something there, and so on.
I do not mean someone intelligent, but the nature of
Nature, okay, someone. No. So anyway, that's my perspective, but it's very hard to prove those things. Okay. Okay. So what we will do, because we are running out of time, I will now shift to the board, and what you'll see in a very quick analysis in a dynamical systems perspective, that not only will you be able to see this behavior coming out, you will also see something very interesting that will ultimately explain the question that I started the lecture today. Okay. So I will shift to the board, and actually I don't think we need this anymore. So can I just remove this, and then can we just pull this up? Okay, so we'll set up the problem. Remember what we've been talking about is we've been talking about two oscillators. One is the circadian clock. Okay, so imagine that you have a circadian clock, and with time you have these oscillations. So for the moment, just imagine that we have these very clean sine wave oscillations. Over the next 2 days, I'll tell you that it's not clean sine wave oscillations, and I'll also tell you tomorrow what exactly these oscillations are, meaning what is there on the y-axis, okay? So for the moment, take my word for it that it's some sort of gene expression. You don't have to understand what this means now, you'll see this tomorrow. But for the moment, just assume this is a sine wave, okay, with some frequency. So for the circadian clock, let's call the phase. So I'm assuming all of you have heard this word phase. So arbitrarily, we will define this peak as phase 0, okay? These are equal to 0. And for the circadian clock, I'm going to describe it with the phase variable, talking about is the day-night cycle. Is the day-night cycle, and the phase of the day-night cycle I'm going to describe with phi. Okay, and once again, imagine this is also a perfect sine wave oscillator, potentially with a slightly different time period. Yeah, and also once again, quite arbitrarily, you can define this to be phase zero. Okay, so for both, I've defined the peaks to be phase zero. Okay, so this one, what we will do is we'll say that the phase velocity d theta by dt we'll call just omega. So this is simply the angular velocity of this phase variable, and for this phase variable d phi dt we'll call this capital omega. Okay, and note that capital omega and small omega are not the same. Okay. And also remember that these are what are called the endogenous frequency. Okay, meaning when there is no coupling. Endogenous or free running. Okay, that's the word that is used in the literature. Free running or endogenous. So now let's introduce a model of coupling. So let's introduce a model of coupling. And this is by, it's quite old, this is by Ermentrout and Rinzel in 1984. So this is a simple model. It was in fact the first time when it was published. It was published in the context of firefly rhythms actually. Okay, and how many of you have seen this textbook by Steven Strogatz? Yeah. Yes, very good. Some of you have. So this analysis is straight from Strogatz. Okay, if you want to look this up. So let's see what the model says. So theta dot, which is my angular velocity for the circadian clock, is omega. That's my endogenous frequency without any coupling. And let's write down a simple coupling term, which looks like this: sine phi minus theta. Okay, so let me call that equation 1. So what does this model say? So think about it in terms of two athletes who are running on a circle, for example, right? So theta describes the circadian clock. This one phi denotes the day-night cycle. So two athletes running on a circle, and we want to understand when they will overlap each other, when they will lap each other, and so on. Okay. So what's happening over here? If you notice, if this term is positive, right? So if this term phi minus theta is greater than 0, then you have this term, you have a positive term over here, right? So omega plus something, right? So your circadian clock is speeding up, the velocity, angular velocity is increasing. So if your day-night cycle is going ahead, your circadian clock is increasing its speed to try and keep up with the day-night cycle. Okay? And if your, and the other way around, right? So if you have this thing going negative, then your circadian clock is slowing down to try and keep up with the day-night cycle. Okay? So the model is very simple and clear, right? Okay. So now we'll just define the phase difference phi, okay, this phi as capital Phi minus theta. Okay, so if I just differentiate this quantity, then I'll have d phi dt is equal to. So what will I get if I differentiate d phi dt? So I have this one here will give me capital omega, right? And then I have theta dot, which from equation 1, I'll put this and we get minus a sine phi. Is that okay? So follow what I'm doing, right? I might make some mistakes. Okay, so typically in the sense of a physics equation, we want to non-dimensionalize it. So I won't spend too much time here, but let's just say let's non-dimensionalize by defining two new variables. Tau is AD and mu is omega minus small omega by A. And you can work this out. It's quite trivial to show that this equation now will become phi dot is equal to d phi d tau is just mu minus sine phi. Okay? Everyone with me? So what we've just done is we started out with two simple equations for the two different oscillators. We construct a model, a very simple physically motivated model for how these oscillators might be coupled. Okay? From that, we've constructed a dynamical equation for the difference in the phase angles between the two oscillators. Okay? And that's the dynamical equation for the difference. Okay. So now in this situation, again, think of these two as athletes on a circular track. Okay? From this discussion of entrainment that we just had before, what do you think would be the condition for entrainment here? What would phi dot need to be so that you have the two athletes running without sort of lapping each other? The phase difference needs to be constant, right? Fantastic. So what does phi dot have to be? Excellent. Great. So for entrainment, you need phi dot to be equal to 0. Okay, so that's the condition that we are looking for. And what we are going to do next in the next 5 minutes is to see where and what, under what conditions will phi dot be equal to 0, where will these solutions be stable, and so on. Okay? All right. So to look at that, a very standard way of looking at it is what is called looking at the flow diagram, the flow lines. Okay? So what we are going to do is we are going to look at 3 different scenarios. Notice that in our equation we have this one parameter here, that's mu, which is mu is this quantity here. The difference in the uncoupled frequencies divided by the coupling constant A. This is some coupling constant, right? Okay. So let's see what happens in the simplest case when mu is equal to 0. Okay? So what I'm going to draw is a diagram where the x-axis is phi, okay, which is my phase difference, and the y-axis is phi dot. Okay, when mu is equal to 0, in this equation you can see phi dot is just minus sine phi. Yeah. So what will this curve look like? It will look like this. Yeah. And what you can see is that here, for values of phi, and I'll tell you why I'm drawing it this way. You have three values of phi where phi dot is equal to 0. Yeah, remember that what we want to look for are conditions where phi dot equal to 0, because that's going to give us entrainment. So now notice, if your system started off somewhere here, for example, at this value of phi, what we want to ask is, will it entrain? Okay? If I start here, in which direction will phi move? Right or left? I hear some people are saying right, some people are saying left.
Some of the second years we get it right.
And then who will get it left?
No, right? I mean, correct.
Yes, okay.
The second years we thought this.
Right. Okay, so you can see what's going on here. It's very simple. Your phi dot is negative over here, right? So phi dot negative means that phi is trying to reduce. Yeah? So your arrow will go like this. And then if your point was somewhere here, it would go like this. Yeah? Because your phi dot here is positive, which means phi is trying to increase. So phi is trying to increase here. Yeah? So this is called what is called a stable fixed point. Okay? So when you set phi dot equal to 0, what you're looking for is what is classically called, you're looking for fixed points. Fixed points means where your velocity is going to 0. Yeah? And here, this is a stable fixed point. Whereas these guys, the ones I drew in open circles, these are unstable fixed points. And the reason is also very easy to see. Here, this is positive, so it's taking it away. Here, it's negative, so it's going that way. Yeah? So if you have a point here, even the smallest fluctuation is going to take you away from this. So in this condition, where mu is equal to 0, and remember, what does mu equal to 0 mean? So mu equal to 0 implies that capital omega is equal to small omega. Yeah? So in this condition, you have only one stable fixed point, which is this stable fixed point is phi star is equal to 0. Okay? So what that means is, if my endogenous circadian clock's rhythm has exactly the same frequency as the external day-night cycle rhythm, then what will happen is you will have entrainment only in a condition where phi star is 0, meaning the two oscillators are oscillating like this, and the other one is also oscillating like this, okay, where the peaks are lining up. Okay, so there's no phase difference between these two oscillations. Okay. Let's see when 0 is less than mu is less than 1. So what does the picture look like now? So if 0 is less than mu is less than 1, you have some positive value of mu here. What it means is it's going to shift up the sine curve, right? Sine goes between -1 and +1. Now you've added a constant term over here, which means it's going to shift up that entire plot, right? So what it will look like now is something like this. So notice what is happening. As I start increasing mu, this line starts getting shifted up and up, right? So at some point, it will touch here at only one point, and then when it goes up more, there are going to be no more fixed points, okay? This in dynamical systems theory is called a critical transition, okay? Anyway, so we won't get into all of that, but notice what is happening here now. Here is one fixed point, yeah, where phi dot goes to 0, and here is one more fixed point where phi dot goes to 0. Okay? If you do the same analysis, you'll see that here your directions look like this, right? Because here phi dot is negative, so your flow is towards this point. Here phi dot is positive, so the flow is towards this point. So this is a stable fixed point. Okay? And what is the value of this fixed point here? You can get that simply by setting this to 0, right? So you have at this point sine of phi star is this quantity here. Okay, so I will come back to this point. This in this single equation hides the idea of chronotypes or why some people wake up much later in the morning and why some people wake up much early in the morning. Okay, so this I'll come back to this. Okay, so hold on to this part over here. So this is called the phase of entrainment. But let me get back here. And finally, you have this case where mu is greater than 1. What would the curve look like over here? So how many fixed points over here now? Zero. Exactly. Very good. So your curve will now look like this. There are no points here where phi dot is equal to 0. Okay. What that means is now, if your mu is significant, is greater than 1, which means the difference between these two frequencies is quite large, you will not have a system where these two athletes are ever able to keep up with each other. Okay, one will constantly get lapped. Okay, so there is no entrainment in this situation over here. Okay, so no fixed points. Yeah. So clearly you can see that mu, the value of mu is setting where, and sorry, how many fixed points you're going to have and whether or not you're going to have any fixed points in the first place. Okay? And of course, if you make mu negative, the argument remains exactly the same, the curve will only shift down. Okay? So just one last thing. Where is the eraser?
On the
So I'll just end in the next few minutes. So the final thing that we want to show is that at entrainment, so if we now plug in this quantity here into equation 1, okay, remember, where is our equation 1? Yeah, we have equation 1 up here, okay. Now at entrainment, if you plug this in here, what you will have is theta dot is equal to omega plus A, right? And sine of phi is that quantity over there. So you can see that theta dot will be just capital O. Okay? So this goes back to the idea that even if you started out with these two oscillators, which have different frequencies, now that they have entrained, your circadian clock is also going to run at the same frequency with which the external day and night cycle is driving. Yeah? So this is exactly the same experiments you saw with the mice. There was a question why in the mice, when you have the 12-hour cycle, why do you see the oscillations going at exactly 24 hours? This is exactly why. Okay? So let's finally end with this thing here. So this quantity here, it's called the phase of entrainment. And what this is saying is that because you have a non-zero phi star at entrainment, what will happen is that your two oscillators will look like this. So let's say your circadian clock is going like this. Your day-night cycle might be going like this. So that could be for one person, right? For another person, it might be going with a slight phase shift. It could be doing something like this. Because notice, in this phase of entrainment expression, you have this quantity here and this quantity here. These are the endogenous frequency of the circadian clock for me, for you, and so on. And it turns out it could be because of genetics. People have shown that certain mutations can cause differences in this quantity between different individuals. So if this quantity is different between different individuals, you can see that the phase of entrainment is going to be different, which means that for person 1 versus person 2, even if these rhythms are going at 24 hours, which is what this equation is saying, you will have these differences with respect to the day-night cycle. This is the day-night cycle. So for some people, when, for example, if you define the phase 0 as, say, afternoon 12 p.m., let's say this is 12 p.m., then for some people, their clock will be at this point, while for others, their clock might be at this point. Okay? Is that clear? Is there any questions? Okay, so we are done. So yeah, just one final thought. So basically, this is what leads to something called chronotypes in humans. Each of us has a 24-hour clock, but that clock is showing different times with respect to the day outside. Okay, and that's what leads to these differences between each of us. So tomorrow, what we will do is we'll come and see how do we actually measure these things in humans. What are exactly the chemical oscillators in our cells that give rise to these oscillations? How do we measure it? And we'll take a different approach tomorrow using ideas from linear algebra to see how you do inference on this kind of systems. So we'll start today. Yeah.
What is the range of cycle till now that has been discovered?
Yes, so it's there is something called a 180-degree phase rule, which means the cycle times for each individual range from I think -6 to 6, so our overall range of 12 hours. So on average, it's 24 hours, 10 minutes, but then you can have a ±6 hours in the phase of different individuals. So 12 hours.
In terms of different not human, but animals and especially what is the range of cycle that has been discovered? Is it still related to 24? Because we can choose only to certain time.
Exactly.
What is the range that we have now?
Not exactly sure. I know that in humans the range is about 12 hours. The total range, the dispersion of this small omega, the variance is about I think 6 hours or something. Yeah, but in general they say that there is this so-called 180-degree phase rule, which suggests that whether it's mouse or humans or so on, this dispersal will be about 12 hours. But the mean can be different. For mice, it's 23 hours. For us, it's 24 hours, 10 minutes, and so on. Yeah.
The likelihood of the circadian rhythm being adopted from the day and night cycle is less because you said that it is intra-species in humans there's a lot of difference
for the day-night cycle to create this, right? What people have shown is that it's extremely simple to put some chemicals together, which will then start oscillating. Okay, we'll see that tomorrow. These are called famously was discovered by a guy called Belusa. So it's very easy to put together a bunch of chemicals and have them start to oscillate. So it's more likely that it was just there, just by chance, bunch of oscillators, chemicals that came together, and then it started uh, getting entrained to the daylight cycle. But again, there are no 100% definite answers to this because it's impossible to know what was happening millions of years back.
Thank you
Yeah.
very much,
Okay.
sir.
Yeah.
If you guys have any more questions, sir will be there outside. You may ask your questions. And thank you very much, sir. We have a photos of volunteer, Varsha. Well, thank you very much for covering today's event. And I hope to see you all tomorrow at the same time. Thank you very much. If you have refreshments kept outside, please do have. Thank you very much.